let's get started. So today we're going to talk about centrifugation, standards, dilutions, and spectrophotometry. Spectrophotometry. Say that 10 times fast. All right, so it's going to be a little bit of a hodgepodge, but this is all going to tie in, except for centrifugation, into today's lab. All right, so centrifugation, it's the process for separating molecules by size, shape, or density using centrifugal forces generated by a spinning rotor. If you've ever been to Disney World and done the Mission Space ride, that's a giant centrifuge. That's how they make you feel like you're weightless and how you have so much gravity on you, because you're literally spinning around in a centrifuge. I only did that ride once, never doing it again. All right. So when you spin, you get all this extra G force on you. And this is used to separate solids, usually solids from liquids, or different density molecules from each other. So a lot of times in the lab, we're going to be using this to separate red blood cells because they're, nice big things compared to plasma from the plasma or the serum by spinning it around really fast. And there are different types. We have our nice little tabletop ones here. These hold microcentrifuge tubes. You can get them where you can hold up to like 24 tubes. They're cute. You get the job done fast. And then you have these big, huge Mamba Jamba floor models. And depending on the type, you can have ones that just do like the 50 ml conical tubes, or you can have ones, and they just, just good for separating, large amounts of things. And then you can have some that are super expensive that can go to insane speeds. And that's usually for analytical preparations. Those, when you balance your tubes, you have to balance them on an analytical balance to like the thousandths of a gram. Otherwise, it gets upset with you. I had to use those in grad school. It's highly annoying. And you have to keep the rotor in the freezer when it's not in use, because the friction of it has to be cold. Otherwise, it gets too hot. So those are fun and expensive. No pressure. Like the rotor itself is $20,000. No pressure at all. You will not be usually using those in a clinical lab. Those are more for research. All right, so the principles of centrifugation is that the denser components of your mixture will migrate away from the axis of the centrifuge, meaning the bottom of the tube, whereas the lighter components, which is usually are like plasma components, will migrate towards the top. So that way the plasma is at the top, the red blood cells and other cells are at the bottom. They're nice and separated, everyone's happy. And we use centrifugation to increase the gravitational force on the test tube to rapidly and completely separate the mixture components, because you can leave a tube sitting out and it will eventually separate, but it will take days. Ain't nobody got time for that. So we use the centrifuge to do it in like 10 minutes. All right, it's just way faster. All right, so when you centrifuge something, the densest components are at the bottom, and it's usually as a pellet. And the least dense components remain at the top, which is called the supernatant. And then when you're actually doing like plasma, you actually get this nice little white layer called the buffy layer, and that's made of white blood cells and platelets. But if you're doing a serum tube, all those white blood cells and platelets are actually trapped in this pellet in like the little fibrin clot. So there's no buffy coat in serum samples. I made this picture myself. I'm very proud. So, All right. So the rate of centrifugation is specified by the angular velocity measured in revolutions per minute, or acceleration, which is a little g, not a big G. And a lot of people think, oh, RPMs and G are the same. They are not the same. It is, you have to convert between them, and that is dependent on the radius of your rotor. So a shorter rotor is going to have a different rotations per minute in terms of G than a much bigger rotor. All right, so in general, bigger rotor, you're going to get more gravitational pull on it at the same revolutions per minute. So the settling velocity is a function of the size and shape, so bigger things will settle faster. The centrifugal acceleration, i.e. how fast is it going? The volume of the fractions of the solids present, the density difference between the particles. So the less difference there is between the particles you're trying to separate, like if there's just the tiniest bit of difference, you're going to have to spin that longer and faster to get them to separate away from each other. And that's why you get those really big analytical centrifuges, because you're trying to separate things that are very close together in density. Whereas red blood cells and plasma, those are pretty different. So you don't have to have the big fancy huge rotor. You can have the little tabletop centrifuge and it will get the job done in a few minutes. And then the liquid viscosity. If it's really thick, it's going to take more time to get through, to get your solid particles through that viscous liquid. It's kind of like going through water versus going through honey kind of thing. If anyone's ever seen vitamin E, it's like super viscous. I had to pipette that a lot. It's gross. All right, so you have your cell extract. It's got all your little goodies in it. And for our purposes of our illustration, big things are more dense, little things are less dense. And you do a low speed spin, and the big things are going to go to the bottom and pellet, but you still have all these, the intermediate and the low density things floating around. And then you can, if you do a high speed spin, so apply more gravitational force, then you'll get the intermediate things with the lightest things still in the supernatant. So depending on the speed and the time of which you're centrifuging it will depend on how they are separated. Longer and more acceleration, the more things are going to go to the bottom. No, there we go. All right, so there's also this fun term called relative centrifugal force, and this is based on the mass, speed, and radius of the rotor. So this is kind of like we have our RPMs and we have our gravitational force. This is a kind of a way to combine them. So the centrifugal acceleration experience at the top of the tube is less than at the bottom of the tube due to the angle of the rotor. And you can use this fun little equation to plug in the radius, the gravitational, the G's, and get this angular velocity thing. I'm really not going to ask you to do this, so you know. This is something you do like once a year when you get a new centrifuge, maybe. All right. Nope. Did it? Yes. Okay. Things being slow. All right. So preparative centrifugation. I had to do this in real life. It's fun. So in this example, preparative centrifugation is used to separate or recover one or more components from a sample from a mixture. So like if you wanted to collect the white blood cells for a DNA extraction, or you want to separate plasma for chemistry assays, those are all preparative centrifugations, but you can also go like one step further and get like start separating by cell type, which is fun. So in this example, someone has taken a liver, they've put it in some buffer, they've homogenized this, which basically means make liver goo. And then they filtered it with some gauze, and that basically trapped all the cellular components in the gauze because they were too big to go through the gauze. And now they have all this other stuff like organelles and things like that, anything that was released by the homogenization process. And they can do serial centrifugation to separate out different components. So for the first centrifugation, they're going to do a low speed for 10 minutes. And that's going to get the really big components out. And then they're going to collect that supernatant and centrifuge it again at a higher speed for a longer period of time. And that will get the intermediate density objects out. And each time they can keep this pellet and do whatever they want with it. All right. So you can keep the pellet and be like, okay, yay, now I have the nuclei. Woohoo. And in this one, they have like the mitochondria and the lysosomes, if they want to do a mitochondrial assay. And then they can take that supernatant again and then do an even harder and longer centrifugation. This is 100,000. That's when you start getting into the big table, like the big floor models, to get the plasma membranes, the fragments. And then you can go even further and faster to get the ribosomes and viruses. You see how these things get progressively smaller each time you do it, and your speed is getting progressively faster and longer. So in grad school, I had to do one where I literally spun it for 24 hours. Like, it was insane. All right. So then we have analytical centrifugation, and this one you're not really trying to collect the parts, you're just trying to analyze them. So these are really cool centrifuges where they actually have a detector underneath these slits in the rotor, and as it gets separated, the detector can actually do like, light and detect the absorbance of things as it's spinning around. So you have a light source at the top, and then you have a detector at the bottom that spans the entire length of the tube, and it will basically make a chromatogram of this is what's in each fraction of the tube as it gets separated. So that's really nifty. So these samples are usually pure, and this is mostly used for basic research. This isn't really done in clinical applications because no one needs that depth of knowledge. You're just going to do like a regular assay to figure out how much of something's in there. But they do exist. All right, so balancing a centrifuge. For some of you, you're like, oh man, I know how to balance a centrifuge. And then for others of you, you think you know how to balance a centrifuge. And then for others of you, you recognize you have no clue. So we're going to talk about it just so everyone's on the same page. So the centrifuge rotor contains slots that will hold tubes. You can either have some that will even hold like micro titer plates, all kinds of things you can get in there, different size tubes. And it's important that the weight is evenly distributed. Because if it is not evenly distributed, you turn that centrifuge on, it is going to sound like an airplane trying to take off. And it will start rocking across the table, and everyone will know that you did not balance your centrifuge. It is very loud. It is very apparent that you did it wrong. And if you do not stop the centrifuge right away, it becomes very unhappy, and you need to stop it right away. So sometimes you need to have a balance tube, and a balance tube is basically any extra tube you add in there that you fill with a similar amount of liquid to the tubes you're trying to balance to. So that way, if you have an odd number of tubes, you can make it an even number of tubes. Or if you have tubes that are very different volumes, you can then make balance tubes that are going to be similar volumes to the tubes you're trying to balance to. So the sample should be matched by both size and volume. So these little examples are here are what not to do. All right. So you should have the same size tube. So these right here are 15 ml conical tubes. And you can see they do not have the same volume. Those are not going to be balanced against each other. You need to get a balance tube for each one of these. So one that's filled to about halfway and one that's all the way full for that to be balanced. If you try to put them directly across from each other, the centrifuge will not be happy. Whereas this one, these both have 15 mLs in there, but those are different size tubes. The tube has its own weight, all right? Don't just think, oh, a tube's a tube. Each tube has its own specific weight. So that's why you need to balance against a particular tube type. All right, so even though these both have 15 mLs in it, they're not going to balance with each other, especially since you're going to have a different size. Usually there's a little cup you insert into the rotor that holds a different size tube. So you'd have to have a different size cup on the opposite side of the rotor to fit the different size tubes, and that rotor itself is going to have a different size, different weight, so it won't be balanced either way. So always have the same size tube with a similar amount of liquid across from each other to make a happy centrifuge. All right, so how to balance a centrifuge. You can never just have one tube. You will always need a balance tube if you're trying to spin one tube. So this little blue circle indicates a balance tube. So those are balanced with each other now, and you put a similar amount of liquid, whereas two, you just put them directly across from each other. I've arbitrarily chosen position 1 and 7 because it made me happy. Now 3, depending on the size of the rotor, you don't necessarily need a balance tube. So this is next level centrifugation balance, okay? If you don't feel confident in this, don't try it, just get a balance tube. But you can do 3 in some rotors. So you notice how I've made an equilateral triangle here. There are 3 holes between each sample. This, now all the weight is evenly distributed in this nice equilateral triangle. If you could not make an equilateral triangle, you have to get a balance tube. So 4, you just do them directly across from each other. It's completely arbitrary where you put them. You could have it in 12 and 6. It doesn't have to be 10 and 4, just each tube has to have a little buddy directly across from it. And then in 5, you make your little equilateral triangle again, and then you have the other tubes you can just have directly across from each other. It all works out. And there's a different way to do 5. You can make a little starfish in some rotors. So there's different ways to do it. Just always make sure if you're doing odd numbers, you have an equilateral, they're equally spaced amongst each other, okay? But when in doubt, use a balance tube. Better to err on the side of a balance tube than to have an unhappy centrifuge. But for those of you who feel confident in yourself and the rotor is capable, you can try to do an odd number. I won't judge you unless you do it wrong, in which case we will all know, because the centrifuge will make a very loud noise. All right. Now we're going to talk about dilutions, simple dilutions first. All right, so a dilution, it consists of adding more solute, i.e. your diluent, to a solution so that the concentration of the solute inside your solution becomes more dilute. It's a dilution. So usually we're diluting patient samples to keep them in the analytical measurement range. Usually our assays only go up so high before they start losing linearity and it gets a little crazy, like the readout you're going to get on the instrument isn't going to match up with the actual concentration. So you need to dilute the sample to get back onto the analytical measurement range or back on the standard curve. And then a lot of times assay reagents are sent as stock solutions that don't have a lot of water in them. And then the lab is expected to dilute it to the final working solution at the concentration that's needed for the assay. And the reason why they do this is because it saves on shipping costs, because you're not shipping a bunch of water, and water is very heavy. And it also saves on storage space in the lab, because you don't need to add the water until you're actually ready to use it. So it just saves a lot of space and money to have these stock solutions that you then dilute right before you use them. And you can have different types of diluents. You can, a lot of times you're going to be using water. You might be using saline or ethanol or acetonitrile. But when you do a dilutions, you notice my little tube has 7 little solute particles in it. This is a 1-to-1. And then I dilute it by adding in more liquid. And I still have 7 solute particles. They haven't magically multiplied or been lost, but now they're more dilute. And then if I add more water, still 7 solute particles. They have more room to breathe now, more space. This is a studio apartment. This is a 3-bedroom apartment. So, all right. So we're going to talk about some of the math involved in doing dilutions and dilution factors. So we have our stock solution, or our sample. This is whatever you're trying to dilute. And for our example, we're going to add 1 ml of our stock solution to 1 ml of diluent. And the thing with dilutions is, unlike what you might be used to in chemistry, the bottom of this fraction is the total number of parts. So for our purpose, so for this, we'd have 1 ml of diluent plus 1 ml of stock solution. That's 2 total parts that we have added together. So we have 1 part of our stock solution and 2 total parts, and that's what makes our dilution. All right. And our dilution factor is whatever the number is at the bottom of our fraction, which is 2 for this example. And that's helpful for calculating the final concentration of something if you had to dilute it to do your assay. So if you had a sample that you had to dilute 1 and 2, then if you wanted to figure out what the actual concentration of the sample was before you dilute it, you just multiply by 2. All right. So the important thing with this is it's always total parts in the bottom here, unlike what you might be used to seeing in chemistry with the little colon, which we'll talk about in sometime in this lecture. All right, so this is considered a 1-to-1 dilution ratio, where you have 1 part of solute and 1 part of diluent. So that is a dilution ratio, and we will always denote it with a colon. Whereas a dilution is going to be denoted with a little division symbol backslash thing. And what's annoying is that even assays made by the same manufacturers, they're written by different people, and depending on their training, they will use this colon, but they will either put it as an actual dilution or a dilution ratio. So you always want to like double check what they're actually telling you to do in the written instructions, because they might be like, oh, make a 1:5, when in reality, they actually want a 1 like 6. So just don't trust the writers of the assays. So most of them have chemistry backgrounds. All right, so in our next example, we've taken 2 mLs of our stock solution and 2 mLs of our diluent. So now we have 2 parts of stock and 4 total parts. And then we can do some basic math and simplify that fraction. And we still have a final dilution of a 1 and 2 with a dilution factor of 2. So same thing, we just did different volumes. This is where you go like parts. So it's just easier. All right, so with this one, we have our, I've given you a concentration now, 100 milligrams per deciliter of our stock solution, plus 1 ml of that, plus 3 mLs of our diluent. So that's now made a 1 and 4, because we have 4 total parts. And so our dilution factor is a 4, and our final dilution is 1 and 4. So if we started with the 100 milligram per deciliter solution, and then we diluted it 1 and 4, then the final concentration of this solution right here is 25. And if we got 25 milligrams per deciliter as our answer in our assay, and we're like, okay, well, what was this concentration of the original solution? We could just multiply by 4 and be like, oh, it was 100. So usually you're going to be doing like multiplying by 4 if you're trying to figure out the concentration of the sample. Whereas with your like reagents, you already know the concentration because you made it, right? So different uses. Yay, math. All right, so dilutions versus dilution ratio. So this terminology can be very confusing. It's even, as I mentioned, it's messed up on package inserts for assays, even by the same manufacturer. Highly annoying. So how you write the dilution expression is important. So for us, a dilution is indicated by the slash. And so a dilution of 1/2 means you bring the volume up of one reagent up to the final volume, the total final volume with the diluent. So this would be 1 part solute to 2 total parts. So you'd be adding 1 part of diluent. All right. Whereas with the dilution ratio that's indicated by the colon, that means you create the solution. A 1:2 dilution ratio means that you create a solution by adding 1 part of solute plus 2 parts of diluent. So this would actually make a dilution of a 1:3. All right. So at low volumes or like low dilutions, it's going to matter a lot which one you did. At really big dilutions, it's going to be like this minor difference and it's not a huge deal, but it's still bad practice, so don't do it. So the differences between dilutions and dilution ratios become smaller when the dilution is super large. So if you're doing a 1 in 1,000, that's 1 part to 1,000 total parts versus a 1 to 100 dilution ratio. So that's a 1,001 parts total. That's not a huge difference in concentration. Like that's just not, because it's such a big dilution. Yes, in the back. So this number on the bottom is always going to indicate the total parts. So what you're actually adding is going to be 999 parts of diluent to make this 1 in 1,000. Whereas this one, you're actually going to be adding 1,000 parts of diluent, but you're technically making 1,001 total parts. So a lot of times in package inserts, it's written what you should do. So like for reagent 1 and reagent 2, and you're supposed to mix them together, it'd be like add 2 parts reagent 1 to 5 parts reagent 2. And then you'd be like, okay, I'm actually making, actually those are really bad numbers to choose. We're going to say you added 1 part of reagent 1 to 5 parts of reagent 2. So what you're actually making is a 1:5 and a 1-6. Those are the same, but they're written differently. All right. So our dilution factor for that dilution would be 5. No, 6. 1, 5, 6. Yes, because that's what you actually did. This is why, as clinical chemists, we like doing the 1-6, like the 1 dash total parts, because you can just take that number on the bottom denominator and go, okay, multiply by that. Whereas if you do the colon, you have to add those 2 numbers together, and that's where you're more likely to make errors. So that's why we prefer doing the dash to total parts, because that gives you that denominator. Denominator is the bottom of a fraction, right? Yeah, okay, math is not my strong suit. That gives you the dilution factor just by default. Whereas with the dilution ratio, you have to do additional math to get to your dilution factor, which is where mistakes can happen. So our goal in life is to avoid mistakes. People's lives depend on it. All right, so when the dilution ratio or dilution is small, any errors is going to be a much bigger if you accidentally switch them up. So for our purposes, we're pretty much always going to be using this 1 dash total parts thing. All right. But a 1-1 means it's not diluted at all because it's only 1 total part. Whereas a 1:1 is diluted. It's confusing, right? Yeah. This is why we're talking about it. Just remember with the dash, it's total parts on the denominator. Whereas with the colon, it's whatever amount of diluent you're adding. So fun times. All right, so we're gonna do some dilution calculations. So you need a 1 in 10 dilution of a 200 milligram per deciliter stock solution with a final volume of 100 mLs. So how would we prepare the solution? And what's the final concentration of the solute in the 1 in 10 dilution? So first thing we're gonna do, we need to figure out how much stock solution we need. So we want 100 mLs. This is what we want. We want a 1 in 10 dilution. So we're going to multiply those together. And we know that we're going to need 10 mLs of our 200 milligram per deciliter stock solution. And then we need to do a volume correction to get how much diluent we need. All right. Because if you just went, okay, well, I have 10 and then I'm going to add 100. you have now made a 1 in 11, which would be wrong. So you need to correct for that volume of diluent by subtracting out how much of your stock solution you're adding. So we take 100 mLs minus 10 to get 90 mLs of diluent. And a lot of times what you're actually going to do in practice, you're going to be doing these usually in volumetric flasks. I don't remember the name. The volumetric flasky things. They have the little etched line. You can get like, I need a 100 ml one. So you'd add your 10 mLs of stock and then you'd QS, which stands for quantity sufficient, to 100 mLs. So you basically just fill to the line with the diluent and you wouldn't actually measure out 90 mLs. And that's important for some assays because especially if you're using like alcohol as your diluent, alcohol, when it's combined with an aqueous solution, it actually loses, it like expands or contracts or something. Like the volume gets really wonky when you add alcohol to a water-based solution. So you need to QS instead of just dumping in 90 mLs because you won't actually end up with the final volume you want. You'll end up with less. And that would be annoying. So this is why we like to QS to our final volume with the diluent. All right. And then we can calculate our final concentration, which would be multiplying our original stock concentration of 200 milligrams per deciliter times the 1 in 10 to get our 20 milligrams per deciliter. And that's another reason why we like the dilution rather than the dilution ratios. It's just easier on the mathematical end to not have to correct for that later. You just multiply by whatever your dilution is. Whereas with the dilution ratio, you have to like, okay, I got to add the 1 back. So it's actually 1 in 11. It's just more steps. All right, so this one we need a 50 milligram per deciliter working solution. And we need 1,000 mLs of it. And we have a 400 milligram per deciliter stock solution. So we need to prepare our working solution and then figure out our dilution factor. So the first thing we're going to do is we're going to take our wonderful C1V1 equals C2V2 equation. And we're going to calculate how much volume of the stock solution we need. So we need 125 mLs of the stock solution. Then then we need to figure out how much diluent we actually need to add if we're not going to do the QS method. So we're going to do 1,000 minus 125, and we're going to be adding 875 mLs of diluent. And then we can either add the 875 mLs or do the QS. And then we'll figure out our dilution factor. So that's 125 mLs over 1,000 mLs total. Then you simplify that fraction, and that would give you a dilution factor of 8 because it's always that number on the denominator. And I will usually be nice and not give you some stupid fraction that's difficult to simplify because no one needs to have that done in their life. This is not math class. All right, serial dilutions. So we was talking about simple dilutions where you're just doing, okay, I'm taking my stock solution, I'm trying to get to my working solution. With serial dilutions, these are very helpful to get to a very big dilution without making a ginormous volume of reagent. So it's also helpful if you're trying to do like immunoassay titers, so as you can figure out where your titer turns positive or negative. So this is a stepwise dilution in the solution, so it gets progressively more dilute after each step by diluting the previous tube. So usually the dilution factor remains constant throughout the serial dilution, resulting in a geometric progression so that the concentration changes in a logarithmic fashion. This is basically a way of saying, so if we're doing a 1 and 2 serial dilution, which is very common, this denominator is getting progressively larger, but this is just 2. This is 2 squared. This is 2 cubed. This is 2 to the 4th, 2 to the 5th, 2 to the 6th, 2 to the 7th, and so on. So you just keep adding a little exponent to whatever your original dilution is for these progressions. So that's in a perfect world where you're just doing a 1 to 2 serial dilution. You can make it more complicated, which we'll talk about. So these are highly accurate, or no, that's wrong. They're accurate to make highly diluted solutions for experiments and laboratory procedures. But if you make a mistake anywhere in that dilution, that serial dilution, it will get magnified the further you go down. So I wouldn't say it's super accurate, but if you are pipetting accurate, then it's good. But if you make a mistake, it's all downhill from there. okay? So, but it's helpful because it minimizes the steps to get to your desired final concentration, but you're also limited by the minimum volume of your pipette. So our pipettes in the lab only go down to 10 microliters. So any dilution that you're going to do, you can never add less than 10 microliters of your stock because you just physically can't do that in our lab. In other labs, you will have fancier pipettes that can, but not here. All right, so if we're doing a 1 to 2 serial dilution, we start with our stock solution of 100 milligrams per deciliter. We add 1 part of solution and 1 part of diluent. So this is now our slightly diluted, very arbitrary terms, which is a 1:2, which is now 50 milligrams per deciliter, because 100 milligrams per deciliter times 1/2 is 50. And then for the next tube, we take from part A, from tube A. So we're not going back to the stock solution. We are done with the stock solution. We take from tube A and dilute that 1:2. So that we take 50 milligrams per deciliter times 1:2, and that would get us 25. But we could also just take the 1:4 that we've now made, which is the total dilution from the stock solution, and multiply that by 100. So you can either calculate it from tube A or you can calculate it from the stock solution. Just keep track of which way you're going here. I personally prefer just to know that this is a 1:4 and the next one's a 1:8 and just multiply it back to the stock solution. Because if you're multiplying back to tube A, if you made a mistake at your calculation at tube A, you've now messed up tube B as well in terms of like your math and saying what the final concentration is. So I prefer just to go back to the stock solution, but it's up to you. Be confident, you know your math skills. All right, and then for tube C, you take 1 part from tube B, add that to 1 part of diluent. And at this point, this is our most diluted tube. This is a 1:2 dilution of tube B, but the total dilution from the stock solution is a 1:8. And you just multiply by 1:2 5 bajillion times. And that's actually 12.5 milligrams per deciliter is our final concentration in that tube. That's what we're going to be doing today in lab. Yay. It's fun. All right. So what's important about serial dilutions is if you're going to be actually using the components of tube A and B in your like assay, you need to make sure that you've left enough liquid in tube A and B to do the assay, because you're taking stuff out of tube A and B to make the next tube, right? So for this one, at the end of this serial dilution, tube A and B are only going to have 1 ml in them, or 1 part. If you needed 2 parts for the assay, you're SOL here. You didn't make enough. So you need to make sure that when you're planning your serial dilution, that you've planned that there is enough remaining in the tubes at the end for you to do the assay. All right, because this is a common mistake. Whereas tube C will have 2 parts. A lot of times that last little part is discarded just so everything matches all evenly. I'm paranoid and I don't like to discard things, because what happens if you need more later? Then I have to do the entire serial dilution again. I don't want to have to do that. So I like to keep the extra parts until I know I don't need them. All right, so our 1:4 serial dilution, we have 1 part of our stock solution and 3 parts of our diluent. This is a 1:4 total dilution. And then we multiplied our 1:4 times 100, and we get 25 milligrams. We were on tube B by the time we got to 25 milligrams in the 1 to 2. This took less steps. That means less time for error. So if you can do it in less steps, do it in less steps. All right, so then we take 1 part from tube A and 3 parts of diluent to tube B. And we now have a 1:4 dilution of tube A, but a total dilution of 1:16, which is 4 squared. And our final dilution, our final concentration of this tube is 6.25. So we're already way better than we were at C. And then our next tube, we take 1 part from tube B with our 3 parts of diluent. We've now made a 1:64. We are very talented very quickly. So we got that very dilute very quickly, but we only used 1 part of our stock solution to get down to 1.56 milligrams per deciliter. So this is a way to use very little of your stock solution to get to a very low concentration. All right, so at the end of this tube, A and B will have 3 parts in them. So that's good. And tube C will have 4 parts in it. All right, 1 and 10. This is a common one too. So you have 1 part solution, 9 parts diluent. So a total dilution of 1 and 10 for the first tube. So it's going to have 10 milligrams per deciliter as its final concentration. Dilute that again, 1 and 10 to make tube B. And we're now at a 1 to 100 dilution. That progressed rapidly. And now we're at 1 milligram per deciliter. And for tube C, we take 1 part of tube B to 9 parts of diluent. We're now a 1 to 1,000 dilution. And we got to 0.1 milligrams per deciliter in 3 tubes. Yay, us. We're fantastic. All right, so tube A and B will have 9 parts in them at the end, and tube C will have 10 parts at the end. All right, so now we're going to talk about complex dilution. So this, in complex dilutions, it's still technically a serial dilution, but you're no longer just doing, okay, I'm just doing 1 to 2s the entire time. This is where you actually have to pay attention more when you're doing your dilution and not mess up. Ask me how I know that. Messed up a few times. So this one, we're going to start with the 1 and 2. So we have our 1 and 2. So our final concentration of tube A is 50 milligrams per deciliter. And then from tube A, I'm going to take 1 part, but I'm going to do 3 parts of diluent this time. Ooh, I'm tricky. So now I've made a 1 to 4 of tube A, but this is actually a total dilution of 1 to 8. So I have my 1 to 2 from the original tube times the 1 to 4 to 1 to 8. So this time it only took me 2 tubes to get to a 1 to 8, whereas with our 1 to 2 serial dilutions, it took 3 tubes. So, and I got down to 12.5 milligrams per deciliter in 1 less step. Whereas with tube C, I'm going to take 1 part from tube B and 9 parts from of diluent. So now I've made a 1 to 10 dilution of 10 of tube B. My total dilution, ignore that number right there. That's supposed to be an 80. My bad. So I've made a 1 to 80, which actually has a concentration of 1.25 milligrams per deciliter. All right. So the reason you're like, well, why would I do this? This is annoying. This is if you're trying to, usually you're going to do this if you're trying to get to a specific concentration. So if you were like, I definitely want like 1.25 milligrams per deciliter, what's the fastest way to get there? This would be what you would start doing complex dilutions. All right, so an example complex dilution problem. So you need to make 10 mLs of a 0.1 milligram per deciliter solution from a 500 milligram per deciliter stock. So this is really concentrated, and you need a very small volume of a 0.1 milligram per deciliter volume. Your pipette has a minimum volume of 100 microliters. This is relevant to this question because you can only pipette the smallest volume that you can accurately pipette. So how would we make this dilution? So what dilution factor is actually needed? What number are we trying to get to, right? So to do this, we're going to take our 500 milligrams over our 0.1, and that's a 5,000. So we're trying to get to a 1 in 5,000 dilution. So if you're being really just like, oh, I could just take 100 microliters of my stock solution, how much volume would you need? You'd be making like 5 liters of your solution when you only need 10. You only need 10 mLs. You don't need 5 liters. So this is why we do these complex solutions. So we don't have to make a ton of extra volume. But then you're like, okay, well, what do I do with it? Do I just toss it down the drain? Oh, wait, I can't toss things down the drain. I have to pay for environmental health and safety to get rid of it for me. So we don't want to make more than we need. So we don't want to just do a straight 1 in 5,000. It just makes way too much solution. So there's more than one way to do this to get to 1 in 5,000. So we could do it where we do a 1 in 5, and then we do several 1 in 10 dilutions. So that's 4 dilutions we had to do to get to our 10 mLs of solution. So with this one, I took 100 microliters to 500 microliters, total microliters, and then I did 100 of that to 1,000, 100 of that to 1,000. And then at that point, I was like, okay, now I can go big to get to my 10 mLs. So that's one way to do it. This took 4 dilutions. Nothing wrong with that. If you wanted to go a different way, you could do a 1 in 2 initially, and then do a 2 1 in 50s to get to your 10 mLs. Nothing wrong with that either. That took 3 dilutions, less likelihood for error. And then you can do it where you do a 1 in 50, then a 1 in 100, where you're always still having your 100 microliters because that's as low as your pipette can go. You made 5 mLs and then 10 mLs. So this one you made 15 total mLs of volume. This one you made 15.2. And this one you made 12.5. So either one, any of these work. Usually in the lab, you have one, like there'll be written instructions. Like if you're trying to get this one, this is the way we want you to do it. But there's more than one way to skin a cat to get to some of these dilutions. I'd probably do this one just because I don't like doing a lot of pipetting. All right, so some tips for serial dilutions that I hope you practice today. So in your racks, you usually have some extra spaces. Make use of those because it's very easy for you to get distracted and to lose track of where you are. So move tubes as you finish pipetting them. So I like to have at least one empty spot to the far left of my tube rack. And as I pipette, I move a tube over to that empty spot. And I just keep moving the tube over to that empty spot so that I don't lose track of where I am. I also will talk out loud to myself. I have no shame. That might be distracting or to your classmates, but you can also, on your next tube, if you have an available hand, I like to keep a finger on the next tube. So that way I'm like, okay, I'm going for you next. So if you're just do something to keep track of where you are, because it is very easy to get distracted and be like, I don't know where I am in the dilution. Which right now when we're using dye, it's pretty easy to tell because it's colored. But when we start using things that aren't colored, things can go, you can get distracted and mess up where you are. And then you have to start over again when you realize that you've done it wrong after you've done your standard curve and go, oh, darn, that does not look the way it's supposed to. You might not always have time to redo it. So have some mechanism in place to avoid making mistakes. All right, so now we're going to talk about standards and calibrators. Let's see if we can get through that. All right. So standards are used to determine unknown concentrations by comparing the signal of the unknown to the standard signal. So these signals can be anything like absorbance, fluorescence, light scatter, anything. For most of our applications in our programs, usually going to be a lot of absorbance. So standards are, these are 3 words, 3 little characteristics I want you to know. They are pure, they are weighed out, and they have a known concentration of your analyte of interest. So your analyte of interest is any substance whose chemical constituents are being identified and measured. So this is like your glucose and your glucose acid. You will have a glucose standard. You know exactly how much glucose is in that glucose standard because that is what you are comparing your unknowns to. So the stock standard is a concentrated form of the standard, and then you dilute it to make your working standard. So this is where you make your working standards to make your standard curve. And these working standards are at a concentration that's appropriate for the assay. So you might have a stock standard that's 1,000 milligrams per deciliter, but you only need to be like 200 milligrams per deciliter and below. So you dilute it to the concentration that you need. So, all right, so there are different grades of standards. You have the pure American Chemical Society limits that are stable, anhydrous, and soluble. These can be used to make solutions of accurately known concentrations. The problem is these are made by chemists. They don't have a lot of the biological analytes that we care about. So they don't always have what we need. So we don't use them. We use them when we can, but if they don't have it, we go down to the NIST certified standard reference materials. And NIST stands for the National Institute of Standards and Technology. And they don't necessarily have the purity of a primary standard, but they have been carefully analyzed and certified for use in the clinical lab. So they're the best we have for some of these biological analytes. And we do a lot of our biological testing on them, using those as our standards. And then we have secondary standards. These are your working standards that you diluted from your stock standard. So these have known concentrations relative to the primary standard that we have, or the NIST traceable one. And we use these to generate our standard curves right here for calculating the concentration of the analyte in our unknowns, or we can use them as a single point standard. We can also have calibrators, and calibrators are different than standards. So standards is one analyte in solution. A calibrator, these are often multiple analytes combined into one solution that you can just plug onto your instrument. And it's like, okay, well, I know the concentration in this for this analyte's X and for this analyte it's Y. So you just basically can have one tube of material on your machine that has everything you need in it. Just save space. So these are used to calibrate the analytical instruments, and they're used a lot. All right, so our single point standards, this is where you have you measure the signal of your secondary standard and then use that to determine the concentration of the unknown. And this is based on Beer's law. I realize I'm not going to have enough time in this lab to cover all this. All right, so Beer's law is helpful in setting up the relationship between signal and concentration. And Beer's law is where absorbance equals A, B, C, where A is the extinction coefficient at a particular wavelength of your analyte of interest. B is the path length that the light is traveling through, and most spectrophotometers is 1 centimeter. And then concentration is your concentration of your analyte of interest. And since the extinction coefficient stays the same because it's unique to that analyte at a particular wavelength, and the path length remains the same because you're not switching between spectrophotometers, you can basically make the assumption that only absorbance and concentration will vary, and they will vary in proportion to each other, which is where we get to this fun little equation here. So the absorbance of the standard and the concentration of the standard equals AB, and the absorbance of the unknown and the concentration of the unknown equals AB. So using mathematical logic, we can make those equal to each other and then solve for the concentration of the unknown. And then we get this nice little equation where the absorbance of the unknown times the concentration of the standard divided by the absorbance of the standard is your concentration of your unknown. Yay. And that's how you calculate your concentration of your unknown from a single point standard. All right, so what's the concentration of the unknown sample? So we have 4 grams per deciliter standard. We have our unknown. We have our absorbances that are recorded, and then we can do math. Plug it into our fun little equation. I end up with 2 grams per deciliter of our unknown. I made this nice. This is literally just 175 times 2 is 350. That was nice for this one. That's not real world. So, but this assumes this only works if the standards have a linear relationship between concentration and the signal output. So at higher concentrations, sometimes this relationship gets non-linear. And at that point, you have to like cut off your standard curve and be like any number, any concentration greater than like 100 for this assay, I'm going to have to dilute my sample because it won't be on the standard curve. All right, so our multi-point standard curve, this is where you have at least three working or secondary standards to make your standard curve. If you have 2, your line is always going to be perfect because it's just a line between 2 points. That's why you have to have 3. So you analyze them in the same procedure as your unknowns. You draw a line of best fit where you try to make it so that the line is equidistant from all the little points on each side. It goes through the origin, and then you do not extrapolate past this highest standard because you don't want to make assumptions. And you can then plot the absorbance or signal of your unknown on this nice little curve and figure out the concentration of the analyte. Or you can just plug the absorbance into the equation. So these graphs, it is up to you about whether you want the concentration to be on the x-axis or the y-axis. It is up to you. You listen to your heart, all right? But you need to keep track of where it is because the equation of the line, where you plug things in is going to matter. So for the way it's drawn right here with the absorbance on the x-axis, I can just plug my absorbance into x in this equation, and y will be the concentration of my unknown. I know a lot of people prefer to do this because it's easier, because you can just plug your thing into x, the absorbance into x, get y, yay. But a lot of times you're going to see it where the absorbance is on the y-axis because it just makes a prettier graph. But in that case, you have to plug your absorbance into y and then solve for x. That's fine too. Just keep track of what you're doing. All right. So we're going to talk multi-point standard curves, and we'll talk about spectrophotometry in lab. So to calculate the unknown, you will plug your absorbance into your curve, into your equation, and solve for y. And you just want to make sure you keep track of which way you're doing it. I won't judge you either way. Just make sure you calculate right. All right. All right, so rules for plotting multi-point standard curves. You must plot at least 3 standards. If you plot 2, it will always be a perfect line, and that does not help you. It must go through the origin. You need to choose a range of standard concentrations that cover your expected range of unknowns or the published analytical measurement range. That's usually provided by the manufacturer of the assay. Do not extrapolate the line. Stop at your highest standards. And you should have an equal number of points on each side of the line that are equidistant from the line. So here we go. Drawing time. Absorbance, concentration. So I have my little lines here. Because in a perfect world, they'd all be on the same line, right? If you had beautifully accurate pipetting. This is not the real world. So when you extrapolate your line, when you draw your line, you want to make it so that these points are all kind of equidistant from the line. So it's like, it's not favoring one side. So that there's more points on one side of the line versus the other. You want to like balance it out so they're all evenly distributed across the line. You want to make sure that you are in the same units as the reporting units for the patient sample, so you don't make mistakes when you're reporting. And you want to label both axes completely. I am a pet peeve that if you're doing absorbance, you must put the wavelength that you're at. All right. If you don't put wavelength, I'm going to dock off point, as well as putting the units for your analyte concentrations, like milligrams per deciliter. All right, where we want to be. So we're going to pick up where we left off in the lecture with spectrophotometry. We won't spend too much time on this because we want to get started on our lab. All right, so some things about light. So light is both particle and energy traveling through waves in space, and these waves can be described. Anyway, these waves can be described in terms of wavelength or frequency. So wavelength is the distance between the peaks or the troughs of the wave. So here we have wavelength as the distance between the peaks, or you can do it right here with just the troughs. And that distance between is the wavelength. And the frequency is the number of peaks or troughs that pass through a particular point in time. So if we were just looking at this one point in time, as the light came through, you'd count how many peaks came through in a certain amount of time. So fun fact, if it is a high frequency, it's usually blue light, where if it's a slow frequency, i.e. it's longer, it's red. I always get that mixed up in my brain. I don't know why, but longer is red, shorter is blue. All right, so humans, because we are weaklings, we can see a very narrow range of wavelengths called the visible light, which is between 750 and 400 nanometers. And this encompasses all the colors of the rainbow that we can see. And each particular wavelength has a color associated with it. So as I said, longer wavelengths are red, shorter wavelengths are blue. This picture is somewhat, I feel like, confusing because this is actually the increasing wavelength. Because its distance between the peaks gets longer, whereas the frequency is higher for blue light. It's a little confusing. Does it not like you over there? No, I don't know what the problem is. I do not know how to troubleshoot that. I'm going to hope that once it warms up, it turns on. Let me know if it doesn't. All right, so spectrophotometry, this is basically us looking at the absorbance or transmittance of light through a substance, because the substance will appear a particular color because it absorbs the visible light corresponding to its complementary color. When I realized this, my mind was a little blown. So if you see a blue object, something that looks blue to your eye, it's because it's absorbing all the orange light. So all the blue light is transmitted through the object. Did not think that was true. So it seems counterintuitive because what you're observing is not what's actually being absorbed by the object. It's what's being transmitted. So you look at the opposite of the little color wheel, which reminds you of elementary school art classes. And we see that if something is appearing as blue, it is absorbing the orange light. So fun. All right. So many components of a solution can absorb UV or visible light. So it can be used to determine the present state or concentration of the substances. So we will typically measure the absorbance or transmittance of light through our sample between 200 and 900 nanometers. And so if we have this nice little red sample here, it is actually going to be absorbing green light. It still just boggles my mind. I do not know why that's not working. Let me see if I can get Kara. I'm just going to check Kara for all technological issues. Versus absorbance. So a sample that has absolutely nothing in it, like basically pure water, that will transmit 100% of the light that hits it. So all the light will go through it. All right, and a sample that has something in it will absorb some of that light, depending on what's in it. It will absorb different wavelengths, and you can measure that with our fun detectors. They're usually like photomultiplier tubes. And you'll get a nice little graph. And as concentration increases, the transmittance will decrease. Because imagine you're in a lake, you kick up all the dirt, you're not going to see as much light through the water, right? I don't understand why your head's underwater in a lake to begin with, because that's disgusting. But the more dirt that's kicked up, the less you're going to see, the less light that's going to be transmitted through the liquid, whereas it will be absorbing more light. So as concentration increases, your absorbance goes up, but your transmittance goes down. And there is a relationship between that, which is absorbance equals the negative log of the transmittance. Yay. So there's a logarithmic relationship between transmittance and absorbance. So an absorbance of 0 corresponds to a transmittance of 100%. An absorbance of 1 corresponds to a transmittance of 10%. And then we're going to talk about our fun little spectrophotometer here. All right, in a second. So Beer's law, this has to do with the absorbance of light. We talked about this a little bit in lecture, where absorbance equals the extinction coefficient of your analyte of interest at a particular wavelength. The B is your path length of light, which is usually 1 centimeter. And then the concentration, C is the concentration. Usually in moles per liter. It depends on the units of the molar absorptivity. So since it's in this particular slide, it's molar absorptivity. That's why your concentration's in moles per liter. So all the units cancel out. So you end up in the right units when the calculation's done. But there's an assumption with Beer's law that this is a linear relationship, and Beer's law doesn't apply if it becomes nonlinear. All right, so we talked about this in lecture 2, just a little refresher, that basically absorbance of the standard and the concentration of the standard equals that molar absorptivity extinction coefficient times the path length, and same goes for the absorption of the absorbance of the unknown and the concentration of the unknown. So you can set them equal to each other and then solve for the concentration of the unknown and use this pretty equation. I know some people that just never bother memorizing this equation, they just know how to solve for it, and that's fine too. However you want to do it. I always get confused about which absorbance is supposed to be on the top. All right, so now we're going to talk about the spectrophotometer. So measurement of light at selected wavelengths. So in order to do that, in order to basically measure our analyte of interest at our particular wavelength that we care about, we have to be able to select for that wavelength. So we have our nice light source in our spectrophotometers. And then there's an entrance slit that helps basically choose one beam of light, so you're not hitting all of this light into your prism. And then the light goes into the prism or grating. It's basically fans it out into all the colors. And then you have a slit, an exit slit, that you can change based on how you've set your spectrophotometer to select a particular wavelength, like 500 nanometers. And it will only let light of that wavelength through to the sample. The light will then pass through the sample. Some of it will get absorbed. Some of it will get transmitted. And the detector will pick up whatever is transmitted and convert that into an electrical signal. Then you get a digital readout like 0.500. So, and that's how it's done. It used to be way sketchier back in the day. Now we get digital readouts. All right, so one thing with spectrophotometry is that a lot of times we're going to be using reagents or samples that have color themselves. And that color just kind of muddies everything up. So you need to be able to correct for the inherent color of your reagent or your sample. So this is when we start making blanks. So we're pretty much always going to have a reagent blank because the reagent's always going to have something in it because it's going to be reacting with the analyte. So a lot of times the reagent has like a color on its own. like protein reagents blue. Sometimes it's not visible to the eye, but there's stuff in there. So we always have a reagent blank, and usually the reagent blank is just your zero standard if you're making a standard curve. But the purpose of the reagent blank is to correct for the color that comes from the reagent. And to make the reagent blank, you take, say if we wanted to do an assay where we have 3 mLs of reagent and 100 microliters of sample, to make our reagent blank, we'd add the 3 mLs of reagent, and instead of adding 100 microliters of sample, we'd add 100 microliters of diluent. So that way we just have the correct volume to compare apples to apples. So the reagent's diluted correctly compared to how it's going to be if you actually add a sample, because there's like the volume difference. So, and then you would subtract this absorbance from your actual samples. And a lot of time this is done for you by the spectrophotometer, because you'll put your reagent blank in there and you'll press the little blank button. And that's just the instrument subtracting that number out for you. It's one less step you have to do, because otherwise you'd have to subtract that number from every single sample. And no one wants to do that. The specimen blank, on the other hand, is one you don't always have to do with every specimen. It's only ones that are particularly troublesome. So ones that are colored or turbid on their own. So this would be things like hemolyzed samples, lipemic samples, icteric samples, anything that's not your classic, nicely colored, slightly yellow sample. So anything that's off the beaten path. So the purpose of the specimen blank is to correct for the color of the specimen itself. So in order to make the specimen blank, you would take your 3 mLs of diluent instead of reagent, and then you'd add your 100 microliters of sample. And that's so you're getting the right volume, because you don't want to have to put 3 mLs of sample in there, and that would just be way darker because it'd be just more concentrated, right? Because you're diluting the sample in the reagent. So this basically, by using diluent instead of reagent, you're diluting the sample the same as it's going to be when it's with the reagent. So it just makes everything comparable. And for this, once you record this absorbance, you would then manually subtract it out from the absorbance of your sample with the reagent. So an example, I made this figure too, still very proud. Okay, so for this one, we have our specimen plus reagent, or this is like the actual test we're doing, where we have 100 microliters of specimen and 1 ml of reagent, and we get an absorbance of 0.85. Yay. So our reagent blank is going to be 100 microliters of diluent and 1 ml of reagent, and it had an absorbance of 0.235. So we must have had a pretty colored reagent for it to be that high. And then our specimen blank, 100 microliters of specimen plus 1 ml of diluent, and it's pretty low, 0.035, but that's a pretty significant contribution. So in order to correct to get the actual absorbance of only our analyte of interest reacting with the reagent, we would take our 0.85 minus the 0.235 from the reagent blank minus the 0.035 from the specimen blank, and our actual corrected absorbance is 0.580. Whereas with our standard, we had 100 milligram per deciliter standard. So we took 100 microliters of that standard, 1 ml of reagent, and we got 0.85. Wait, that's the exact same we got for the specimen. Amazing. Coincidence? I think not. It's almost like Dr. Moss planned it that way. And then we make our reagent blank. We can use the exact same one we used above. And we get that 0.235. So to get our corrected absorbance for our standard, we subtract that 0.85 or 0.235 from the 0.85, and we get 0.615. If we hadn't done this correction, we would have thought that they were the exact same concentration because they had the exact same absorbance. But if we actually calculate out the concentration using the corrected absorbance values, we get that our sample is only 94 milligrams per deciliter instead of 100, because it did have its own inherent color that we needed to correct for. And that's why specimen blanks are important, but also because they're time consuming. You only do them if your sample is kind of above and beyond the normal color you would expect for a serum sample or something, because otherwise it's just going to use a lot of time in tubes and materials. So you really only need them if your sample is hemolyzed, icteric, or lipemic.